Alright guys, as promised, it's going to make you a little review on our diffusion, our facilitated diffusion, and our active transport. And another video I'll make you a little review on osmosis, but I want to just start off with this one. So first off, I made a very long portion of the cell membrane. So this area out here, we're going to say this is outside of the cell. And this area down here, we're going to say this is inside of our cell. Okay? That's going to be the same for all of our examples we go over, so at least you have kind of a frame of reference that this would be inside the cell, this would be outside of the cell, and then here's our cell membrane going across, made of our phospholipids. So I'm going to start over here. We're going to start with just regular diffusion, nothing special. Okay. And what I'm going to do, guys, is I'm actually going to draw some lines in here. Okay. And we're just going to use that portion of the cell membrane up to the brown line to just model diffusion. Okay. First off, diffusion, we know it's always going to move high to low concentration. So it's going to move from wherever there's more of something to wherever there's less of something. Also, it's going to move small nonpolar molecules. So let's, I don't know, we'll move carbon dioxide. That's just an example. I know another example we went over in class was oxygen gas, the gas that we breathe. Okay, so both of those, oxygen gas, carbon dioxide, they're very small, they're nonpolar, all right? So okay, so I drew some carbon dioxide, 1, 2, 3, 4, 5, 6, 7 of them outside of the cell. And then I'm going to draw some inside of the cell as well. Okay. So there's our carbon dioxide. Right now, this will be our starting time. So this is where we started out at. Again, we have 7 on this side, we have 3 on this side. First off, we're going to determine which one is high concentration. Is outside of the cell high concentration, or is inside of the cell high concentration with respect to our carbon dioxide? Well, that's pretty simple. All you got to do is count. 7 on this side, 3 on this side. High concentration is wherever there's more, so this is going to be our high concentration. We're going to move from high to low. So what we're going to do is we are going to move through our cell membrane, right through our phospholipids, and the carbon dioxide is going to go inside of the cell. Next, we've got to determine how many of them are going to move. So again, 7 on this side, 3 on this side, add them up, we get 10 altogether. So in order to reach equilibrium, or the same amount on each side, we have to have 10 altogether, so divide that by 2, we're going to have 5 on each side. So we are going to take 2 of them, we'll just take these 2 here, and we are going to move them to the other side of our cell membrane. So there's 1, there's 2, okay. Oops, sorry guys, that was my mistake. So there's our 1, there's our 2, that's those 2 that moved through. So after that, these ones are no longer on this side of the cell membrane. So we moved our 2 carbon dioxides across. We have 1, 2, 3, 4, 5 here, 1, 2, 3, 4, 5 here. We are now at equilibrium. So our big notes on diffusion is that it's going to move small, nonpolar molecules like carbon dioxide or oxygen gas. It's going to go from high to low concentration until equilibrium is reached. And it's able to move right through our phospholipids, mostly because it's nonpolar. It's not going to be repelled by our fatty acid tails in the middle there. All right. So that is diffusion. That's all you have to know for that. Let's go to our next one. Next one, we're going to do facilitated diffusion. Okay. And we'll take that about to right here. So in the first one, guys, this is diffusion. In this middle area, this will be facilitated diffusion. What we're going to move in facilitated diffusion, these are going to be molecules that are much larger or polar compared to our small nonpolar ones that we moved in diffusion. I'm going to move glucose in here. Okay, so let me just make a few glucoses. There's a glucose. There's another one. Okay, and then I'll make, I don't know, maybe one on this side of the cell membrane. Okay, so since these are large polar molecules, we're not going to be able to move right through the phospholipids, right through our cell membrane, as we did with diffusion. Instead, we're going to have to move through something else. So what I'm going to do to help our glucose move across our cell membrane is I'm going to take out some of these phospholipids. Now, in reality, guys, in the cell membrane, they're not going to be taken out. We're going to have things floating in our fluid mosaic. Okay, cell membrane is referred to as sometimes because everything is moving kind of like in a fluid fashion. But we're going to have some proteins in here in our cell membrane, and there's going to be a lot of them. And we're going to make a little channel in the middle here. All right, so that's our protein. Okay, most of the time for facilitated diffusion, it's going to be moved by a channel protein. And what's going to happen here, guys, is once again, okay, if you remember for diffusion, our goal was to reach equilibrium, or the same amount on each side. This is also the goal for facilitated diffusion. We are also going to try to have the same amount, or reach equilibrium, on each side of the cell membrane, whether it's outside of the cell and inside of the cell. We want the same number. So we got 1, 2, 3 outside. We got 1 inside. We're going to have to move one of these. So I'm just going to pull this guy here. We're going to take him, and we're going to move him down our concentration gradient, okay? And we're going to take him to the other side. So now we got 2 glucose molecules on this side and 2 on this side. And this guy, he's no longer there because he moved across. So similarities between diffusion and facilitated diffusion is they're both going to move from high to low concentration. This being high for both of them, and this being low. They both move this way. Things were going into the cell membrane. Now, it always doesn't have to happen that way. It's just wherever there's more, that's where you're going to start with, and you're going to move more to where there's less. So that's one similarity, is they're both going to move from high to low. Another one is that both of these, if you notice, remember we talked about ATP, that's used for energy in our cells and in our bodies. No energy, no ATP is being used in either one of these. All right, so both going from high to low, both do not use energy. The only real difference is this one is going to move through the phospholipid bilayer because these are nice, small, nonpolar molecules. But this one, since glucose is much bigger, it's going to have to move through a channel protein. All right, so facilitated diffusion right through a channel protein. Along with this, guys, along with these being large polar, facilitated diffusion is also going to move ions. Okay, they could be like chlorine with a negative sign, okay, or maybe like some hydrogens with a positive sign. So anything that's an ion or charged, they're going to have some little plus or minus thing there. Okay, that's a lot with facilitated diffusion. All right. So those are both types of passive transport. We'll go over osmosis in another video, which is our last form of passive transport. But there's diffusion and facilitated diffusion. Our only type of active transport, in other words, one that uses energy, is actually exactly called that name, and it's active transport. Okay. And our most famous one is called the sodium-potassium pump, which I'll model that real quick for you guys. It's also going to need a protein, so let me erase some of these here. Okay. And we're going to put protein in. Made this one a little bit different, our protein, okay? So for active transport, I'm gonna be moving two different things on this one. Differences between active transport and facilitated diffusion and regular diffusion is, instead of active transport moving from high to low like facilitated or diffusion did, this one's gonna move low to high, okay? So we're gonna be going against our concentration gradient. In this example, I'm gonna be moving potassium and sodium. So let me put some potassiums here. So on this side here. Okay. Put a few more. And we're also going to put some sodiums. Okay, so in this example, I'll draw one more of these here. Let's just work with the case first, okay? Again, active transport is going to be moving from low to high. Since it's moving from low to high, unlike facilitated diffusion and regular diffusion, we're going to need energy in this one, in the form of ATP. So we're moving low to high. Let's determine with the potassiums where is low and where is high. This side we got 1, 2. Over here we got 1, 2, 3, 4, 5. This is going to be our low concentration because we have less over there. So with active transport, guys, we're not worrying about reaching equilibrium anymore. In other words, we're not worried about reaching the same amount on each side because we're moving from low to high. So in reality, we're probably going to have none on one side and a whole bunch on the other side. So what's going to happen here, guys, is we are going to be moving our potassium and our sodium at the same time. And this is a little bit of a different type of protein. It's called a carrier protein. It usually opens up on one side, and then as it opens up on the one side, it's closed on the other. And it'll actually switch positions and open up on the other side, and it'll spit things out either inside or outside the cell. What's going to happen here, guys, is we don't want sodium inside of our cell. Okay, sodium, think of it as salt. Wherever salt goes, water follows. So if you get too much water inside your cell, it's going to get bigger, 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 and then the cell's later going to burst or go through cytolysis, which is bursting of the cell. So we want to get sodium out, all right? And if you notice here, as we move these, there's these three little indentations here, okay? We are going to have these sodiums go right into our protein, right there, right there, and right there. Okay? And they're going to be moved 1, 2, 3 to the outside of our cell. So they'll go through here and they'll be spit out on the other side. Okay. So here's our 1, 2, 3. They got moved from 1, 2, 3. These ones came over and they went through our protein. Okay? Now, these sodiums have a positive charge. The potassiums also have a positive charge. So as the sodiums are moving outward, again, against the concentration gradient, these little potassiums here, if you notice right here, they're going to be coming, sorry for the color change here, these ones are going to be coming into our cell. Okay? So they're going to come over through our carrier protein and come to the other side. Okay? Whenever all this happens, guys, the big thing is that in order for this to happen, we need to have energy. We need ATP to be added into the equation in order for the potassium to come into the cell and the sodium to come out of the cell. Okay? So looking at the big picture here, guys, just to review. All right, diffusion, this is going to move small nonpolar molecules. It's going to go from high to low concentration. It's going to keep going until equilibrium, or the same amount on each side, is reached. Okay? And then lastly, it's going to move right through our phospholipids. Facilitated diffusion also moves high to low. Still no energy is needed, just like diffusion. Okay? But it's going to move large polar, like glucose, or charged ions, charged particles, through our cell membrane. Okay? But instead of going right through the phospholipids, it's going to use a protein on this one. Okay? So those are your differences. But it's still going to continue until equilibrium is reached. It's still going to go high to low concentration. Active transport is the weird one. Instead of going high to low like those ones did, if you looked here, we had a low amount of potassium on this side, a high amount on this side. It's going to go from low to high concentration. And then it's also going to need energy here. That's active transport. Active transport's the only one that needs energy. It's the only one that goes from low to high. All right. It's still going to use a protein, just like facilitated diffusion, but overall, this one's moving things against their concentration gradient. All right, guys. I'm going to film a video on osmosis as well, but hopefully this one helped you with reviewing those three.